So we're doing a sip of science again. This is the first time in a very long time, a couple of years anyway, that we've done this in person. So it's really nice to have people present again instead of doing it online. So I'm Josh LeBaire. I'm the executive director of the Biodesign Institute, which you see on the various screens all around the room, I see. Okay, we need to get a little closer here. I'm the executive director. So the Biodesign Institute is kind of the premier research institute at Arizona State University. This institute started around the turn of the century, and the focus for our institute has always been solving world problems. And so, unlike some institutes which are focused on just sort of uncovering knowledge, ours is very specifically focused on solving problems. And that lends itself to a different kind of organization. So unlike most places where you would organize scientists by their backgrounds, like a chemistry department or a physics department, we do not have departments at all because you don't solve problems with everybody in a group looking the same. You solve problems by bringing people together with different backgrounds, each one bringing different skill sets. And so we're organized by centers, and each center is focused on a problem. So you'll find centers focused on sustainability, centers focused on diagnostics, centers focused on virotherapies and immunotherapies. Any kind of problem or world challenge, cybersecurity, we'll find we try to build a center there in that area, and then bring in scientists from all over the university. And so I know my center the best, and I know that in my center we have scientists from the schools of engineering, from the College of Health, Health Solutions, from the School of Life Sciences, from the School of Molecular Sciences. I have everything from computer scientists to physicians in the same center. So this idea of bringing people together with different backgrounds is, we think, the way to solve problems. And that's what this institute is all about. So as part of what we do, we felt it was important to share a lot of what's going on with the community. So this is the Sip of Science was sort of created as an opportunity for us to get out and share with people what we're working on. Not necessarily details about our experiments, but more broadly, what kinds of problems are we trying to solve at the Institute? The intent here is really to speak at a level where everybody, who doesn't necessarily have a PhD can understand what we're doing, and most importantly, to give everybody a chance to ask questions. And so the speakers have committed to speaking 10 to 15, 20 minutes, no more than that. And the rest of the time will all be devoted to answering your questions. So please take that opportunity and let's have a vibrant discussion. As I mentioned, we're really thrilled that we're in person. We haven't done that in a very long time. So that's really exciting for us. Okay, and then I'm just going to briefly mention where we're located. So we're currently at the ASU Health Future Center. So this building, if you'd come here 2 years ago, didn't exist. It was dirt back then. The building has just been built. It's just literally been finished about a little bit less than a year ago. And it's specifically to celebrate the connection between ASU and the Mayo Clinic, which is directly next door in that direction. And in fact, some of the faculty at the Mayo Clinic have their offices in this building. And we do experiments here related to the work that they're doing over there. If you go that direction, you'll see a whole bunch of laboratories. You can see that pictured in the slide there. So we have a lot of open space laboratories and people who are working on problems related to human health and problems related to disease. Often have lab space here. Some of the people in my center have lab space here to work on their problems. Over there are also rooms to see patients if we need to collect samples and that sort of thing. And then this side of the campus is, as you see, more of a conference center. And a number of medical societies have held their conferences in this building already, and it's only been here for about a year. Anyway, so that's where you're at. This is a brand new building. This wall here disappears if we needed to, and we have a very large room that we can build if we need to. Okay, so it's my pleasure to introduce the two speakers today. These are both people that I know well. First, I don't know who's speaking first and who's speaking second. So I'll just mention Heather Bean, who did her training at Georgia Tech, and she approaches science looking at very small molecules that travel in our systems called metabolites. And these are little chemicals that signals that they carry information around our bodies. And by looking at patterns of metabolites, you can understand more about a person's health. She's looking at metabolites in the breath, and she'll be talking about how she uses that in the context of the topic today. And then Mitch McGee, who I know very well, he's one of the first people that joined my group when I first got to Arizona. Mitch trained at the University of Texas and then eventually in Pittsburgh, and then back to Texas. He has training both in immunology, which is our body's ability to respond to foreign invaders, and also in proteomics, which is looking at proteins, which are the building blocks of our bodies. So without further ado, I'm going to let them get up and talk about the topic today, and I will sit down. So thank you very much.
All right, I'm live. Thank you so much for having us. So we're actually going to tag team today. Mitch and I have taught undergrads together, microbiology classes, and this is our opportunity to get our duo back together if we did a tag team. Mitch, you're going to drive the
you want me?
stick?
Okay. You want me to stick? Okay.
Sure. All right. So what we're going to be talking about tonight is valley fever. And here's the scientific name for it, which I had to learn how to say coming from Georgia. We don't have this disease in Georgia. It's coccidioidomycosis. Coccidioidomycosis. 3 times fast. So what is this disease? It is a disease that's caused by a fungus that dwells in desert soils. There are 2 different species of coccidioides. That's the genus that the fungus belongs to. There's Coccidioides immitis and Coccidioides possidacei. And both of them can cause disease in humans. It is a CDC reportable disease since 1997, which means that particularly in the states where it's endemic, and I'll show you a graphic on that, if a person is diagnosed with valley fever, that diagnosis needs to be reported to the CDC. It was also listed as a select agent from 2001 to 2013 because it was considered a potential bioterrorism threat, but it can cause disease in otherwise healthy people, which is why it's an important pathogen for us to understand and learn how to track and learn how to diagnose. I mentioned that this fungus lives in the desert soil, and we can see a graphic of that here in the bottom left-hand corner. It grows in the soil in a form called mycelia. This is how the fungus likes to live. Fungi, they make their living by degrading organic matter, so breaking down dead plants and dead animals that are in the soils. But they also happen to be able to switch to a different life cycle whenever they encounter a host that has a nice set of moist, warm lungs that they can replicate in. And they enter into an entirely different life cycle whenever they're inhaled by a host. And we are a host for coccidioides. We can see here in this graphic on the right that by year, we see the incidence number of cases for valley fever. And we can see that incidence has been rising. That could be because we're getting better at diagnosing the disease, although we have a long way to go, which we'll be talking about tonight. But another important aspect that you'll see from this graphic is that Arizona is one of the hotbeds for valley fever. That's the part of the bars that is in orange. In Arizona and Southern California are the most prevalent places where you're going to encounter this desert fungus and inhale it into your lungs and potentially become infected.
Okay, this is part of the tag team. We're switching. So I wanted to show you a little bit different. If you look at the, I'm not sure what's the best way. Oops, go back where we were. This is the organism as we grow it in the lab. You have to have a very special lab to grow this organism because it's actually very dangerous to deal with it in the laboratory environment. We have what's called a biosafety level 3 lab at the Biodesign Institute where we grow the organism. So it's in high containment. Not anybody can just go take a look at it. We have to wear special respirators when we work in the lab because we don't want to get infected. I mean, I've worked with this organism now over 20 years, and I never caught it in the lab, but I think I got it when I moved to Arizona, just like you. But I didn't cure it with, we'll talk about matzo. So again, this has two phases. It's called dimorphic. And so if you look at this, if you look at it in the nature, it grows like a mold on a piece of bread. It's all fuzzy and kind of white, tan. It doesn't look very scary at all. But when it dries out, it forms this thing called the spore, or another, I'm not going to give you the other name, but it's the spore. And we think that when the soil is disturbed, the spores get in the air. And all you have to do is take a breath at the wrong time in the wrong place, and it goes down deep into your lungs. Now, once it gets into your lungs, it changes physiology and grows into this thing that we call a spherule. It's a horrible name, but these are arthrochonidia, these are spherules, and the little bitty beads that break out of it is called an endospore. You don't have to remember that, but just remember that there's two phases. Now, this little bar here is an electron micrograph, is 1 micron. The reason I point that out is this is a lung tissue from a mouse that's been infected, and this whole thing is a spherule. And you have white blood cells in your body that try to kill and phagocytize, eat foreign invaders. Part of the problem is this is one of the phagocytic cells. It's only a tenth the size of the spherule. This spherule is so large that it can't be eaten well. So you have to kill it in a different way. Get down to the next slide. And so this is just another quick review of some of the places where it grows. It was first isolated in South America, the very first case. And the name valley fever actually comes from the San Joaquin Valley. It's not Arizona, Valley of the Sun. But if you look at it, it's in this lower Sonoran life zone, the desert area. And so it grows out in the wild. If you cultivate the ground and enrich it, it doesn't tend to grow in cultivated ground. It likes the desert soils, and particularly around, as you'll see in a minute, around rodent burrows. It's one of the places it tends to gather. And so I'll point this out. This is from 2019, Maricopa County. We're number one. Yeah, so we're number one. We have so Arizona has the most cases, right, of the whole U.S., even more so than California, although they get more of the attention. But Maricopa County, and I did the numbers today for 2019, this the year we have the most complete data. Maricopa County has 61.9% of the population of Arizona, but 71.5% of the cases of valley fever. And those are just the cases that get reported. I don't think we've said it yet, but most of the cases are asymptomatic. You never know you've even had the disease. It's only the severe cases that get noticed. So again, Maricopa County, we're number one. Okay. Whoops.
And so, how do you know if you have valley fever? It's actually really, really hard. And part of the problem is that the symptoms of this disease are very nonspecific. They are mimicked by a lot of other types of respiratory infections. So some of the most common symptoms that you might experience that would lend you to think that you might be dealing with valley fever is that you have fatigue. And not just 1 or 2 days where you're feeling fatigued. It can go on for weeks or months at a time that you're feeling really chronic fatigue. You'll have a cough, but it's not productive. You're not coughing stuff up. You're dealing with fever and night sweats, arthralgia. Erythema nodosum, which I'll show you a picture of in a minute, is an inflammation of the fatty layer under your skin that's very painful. You can end up with skin lesions. And if it's pretty severe, you can end up with weight loss or hemoptysis, which is bloody discharge in your lungs. The problem is that this disease is very difficult to diagnose, and we will talk about some of the reasons why shortly. But really, the way it's diagnosed, the gold standard is you get a sample from the patient's lung, and you can actually grow that fuzzy fungus in the lab. That's the microbiology diagnostic, and you can look at the fungus under the microscope. You can perform tests on that fungus and confirm that what was grown was coccidioides. The other mechanism, because it's very hard to get samples from the lungs, it can be invasive. It's often involving anesthesia and an endoscope going down into your lungs. The alternate approach is to do serology, which means to measure your immune system's response to the fungus. Has it seen the fungus? Can it tell the clinician that your immune system has seen the fungus? And there are lots of problems with serology that we'll be talking about in detail. Now, as Mitch alluded to a few minutes ago, the positive side is most people who encounter this fungus don't actually have noticeable symptoms, or their symptoms resolve on their own before they ever get diagnosed with valley fever. So you might feel fatigued, you might have an unproductive cough that goes on for a couple weeks or a couple months or so, but eventually your immune system is able to take care of that infection and your symptoms resolve, or they're minor and you never seek a diagnosis. So that's the 60% of patients who have had valley fever, but they are actually asymptomatic. Of the remaining 40%, about 30% get acute pneumonia. That's where you have respiratory symptoms and you're dealing with an infection in your lungs. Now, the remaining 10% of people have really severe disease. And this is what we get, a chronic pneumonia, or we get some of these other symptoms that we see here, where the fungus is leaving the lungs and going to other tissues of your body and causing infections there. It can deposit in bones. It can deposit in skin. And we see this person here had a skin lesion that was created by the fungus that migrated out of the lungs. Here's that erythema nodosum and multiforme and nodosum here, where you have that inflammation that's in that fatty layer under your skin that looks like it's causing a rash and is quite painful. And then there are people who have disseminated disease. It can go to, as I mentioned, bone. It can go to your central nervous system. It can go to the brain. And it's typically people who are immune compromised in some significant way, who are at greatest risk of dissemination. But the good news is that most of us living in this endemic area where we encounter this fungus will be infected. We will never know we were infected, and we have immunity to the fungus for at least a good portion of our life after that. So how many of you have lived in Arizona for more than 10 years? Show of hands. Oh yeah, okay. So of those who have your hands up, half of you have probably had valley fever. Cool. There is a 5% chance per year that you've encountered this fungus and actually been infected with valley fever. Yes, you had a question? So Barry's question was, would he have a better chance of getting it today than 10 years ago? Are you based on the incidence rate? It's hard to say. So it could be that the increased incidence that we're detecting could be that we're getting better at identifying some of these folks who have infections. It could be that the incidence rate is rising, and part of the reasons for that would be some of the ways that we encounter the fungus. Thank you for this segue. one of the primary ways that we're encountering the fungus is we have to disturb that desert soil in order to encounter it. I can't pass a valley fever infection to Josh. I can't pass it to my pets. Our pets are often the victims of this disease. They can't pass it to me. The only way you can get this disease is from your encounter with that spore that was in their desert soil. So soil disturbing activities is what puts us at greater risk, and the more soil disturbing activities we do in this area, the more likely we are to encounter the fungus and become infected. So the major risk factors for severe disease: you live in an endemic area. We live in an endemic area. Number one, as Mitch pointed out, if you are engaged in outdoor activities with dust or soil. So even though haboobs disturb a lot of desert soil, that is not the primary way that we are being exposed to dust and therefore being exposed to valley fever. It is primarily by construction. And even if you're living in an area, your house is near a construction zone, if there's fungus in that soil at that spot, you are getting exposed. And then there are certain occupations, so people who work in construction, people who work in archaeology, in wildland firefighting, where they're disturbing the soil to create those fire breaks, and then people who engage in recreational activities that disturb the soil. You're going to be most at risk for encountering that fungus. And then for reasons we don't really understand, people of African and Filipino ancestry are more at risk for developing severe forms of the disease. If you are immunosuppressed, so that could be you have a chronic disease such as HIV that creates immunosuppression, but it could also be the medications that we take to help control rheumatoid arthritis like Humira, or you're undergoing chemotherapy that can create immunosuppression that makes it harder for your immune system to control that fungus when it encounters it. And then pregnancy is actually a form of immunosuppression. And so pregnant people are at a higher risk for developing severe disease.
All right. So again, thinking about Arizona, it's not just humans that get infected. I can tell you what we know is that lizards, reptiles in the desert can get infected. I mentioned that the rodents that dig around in the soil, disturb the soil, they die and they release the fungus and it grows up. So it's all around us. And a couple of things to point out. I've had conversations with the vets at the Phoenix Zoo. It's a major problem for some of our zoo animals, particularly the primates. Out in California, they've seen dolphins with valley fever, that the dust blows all the way out into the ocean, and even dolphins can get it. Seals can get it. And we're going to talk a lot about canines, because if you think about canines in our area, what are they doing? They're smelling the ground. They're breathing those spores right in. And luckily, we have some colleagues at the University of Arizona that are developing a vaccine, and we'll talk a little bit more about it. Did I go too far? But we're going to, they're talking about creating a vaccine that's undergoing testing for dogs. But we're going to take a little break and talk about your immune system. With all the work that has been done with COVID in the past couple of years, hopefully you've learned a little bit about your immune system. You've heard the word antibodies. We're going to talk a little bit more about what they are. So if you look at your, If you look at your immune system, we have what we call the adaptive immune system, the thing that what you really want to recognize. And the whole idea is that you have cells called lymphocytes in your blood that control your immune system. HIV kills all your a specific set of T cells. B cells are the actual lymphocyte in your blood that makes the antibody. Now the antibody, talking about proteomics, antibodies, I think, are the most beautiful protein in the body. They're this Y-shaped molecule that has two arms, and they grip on and hold tight to the foreign protein, the bacterial or the fungal antigen. The protein, and they just won't let go. They hold on tight. And they work with phagocytic cells. Remember I talked about phagocytic cells? They're called macrophages. Antibodies work with phagocytes to help kill the microorganism. But if your microorganism is 10 times the size of your phagocyte, it's a lot harder to do. And so we'll talk a little bit more about your immune system. And the whole idea with the vaccine is that you prime your immune system so that you're ready to engage it. When you first get an infection, you have what's called your primary response. It's typically pretty weak. It takes a little bit of time to get engaged. But if you encounter an infection the second time, you have what's called a secondary response. The response is much higher and much quicker. And the whole idea is with a vaccine is that you're priming your immune system. And again, this is one of the wonderful things about the COVID vaccine is that primes your immune system so that if you see a live virus, you respond real quick and very vigorously. And so vaccines, if you can think about it, it's kind of hard to figure out what's in a vaccine. There's kind of three major ways to make a vaccine. You can kill an organism and grind it up and give it to somebody and hope that that protects them. That usually is not very successful. And we actually tried it in the 1980s. They created what they called a spheral vaccine, a formalin-killed spheral vaccine. It didn't work in humans. It works pretty good in mice, works pretty good in dogs, but it's very toxic. Remember, whenever you get an immune response, many of the symptoms are your immune system engaged to give you fever, pain, swelling. The other way is to create a weakened organism. And you may have heard that some of the live influenza vaccines are attenuated, they're weak. They can't cause disease, but they stimulate a really good immune response. Polio virus, the monkeypox virus are weakened viruses that don't cause disease, but they stimulate a really good immune response. The third way is to create just a small fragment or what we call a subunit vaccine. And this is what currently the coronavirus vaccine is. It's just that spike protein. You've heard about the spike protein. That's what allows it to get into cells. But if you have an antibody to that spike protein, the virus can't get into your cells. If it can't get in, it can't cause an infection. So the whole idea here is that we want to give a vaccine or find a vaccine that can prime the immune system so that if you ever see the live organism, you're protected. And so there is a group at Tucson. Dr. Mark Orbach came up with an attenuated vaccine. He's a very good colleague of ours. And they have a weakened valley fever fungus. And so it's a live vaccine. They're testing it in dogs, and the results look fabulous. And I'm hoping that it will eventually lead to what we call human trials. But it's a big leap to go from animals into humans.
So the best strategy to deal with disease is to prevent it. And that's the whole point of developing a vaccine, but we're not there yet. So when we can't prevent a disease, the next most important thing for us to be able to do is to diagnose that disease. We need to be able to identify what's causing that pneumonia so that we know how to treat it, that we can provide antifungals rather than antibacterials, which are not effective against valley fever. And that's what Mitch and I both work in. We work in diagnostics. In my research lab, one of the main things we work on is developing diagnostics that work by detecting small molecule metabolites that Josh described in our breath, by looking at the chemistry in our breath. The hypothesis that surrounds all of my work is that if you have a different lung pathogen, your breath chemistry will be different if you have Coccidioides versus Pseudomonas versus Staphylococcus aureus. And part of that is because of the metabolites that the bacteria or the fungus is making. Part of that is the metabolites that our immune system is making when it recognizes and fights that pathogen. And the specific chemistry that we identify in breath can be linked to the type of infection that is in our lungs. And we can use this chemistry to diagnose what's causing that lung infection. That's the entire foundation of the work that we do in breath-based diagnostics. So I'm relatively new-ish to valley fever. I don't have decades of experience in it. I started working on it when I came to ASU. And I'll tell you a little bit about the mouse data that we've recently developed to show that we should be able to develop a breath test for valley fever. But the first thing I want to do is just to illustrate that this is real. It's not a pipe dream. We're wrapping up a clinical study now in developing a breath test for lung infections caused by a bacteria called Pseudomonas aeruginosa. This is a pathogen that causes ventilator-associated pneumonia. It causes lung infections in people who have COPD or who have cystic fibrosis, which is a genetic disorder. And in our clinical study, we're finding that with the breath chemistry, we can identify people who have a Pseudomonas infection versus not with 90% accuracy. And that is head and shoulders above our current clinical diagnostic scheme. So seeing that we're seeing success in this bacterial infection, pivoting to the fungus, can we develop a breath test for a fungal infection? One of the primary things that we do before we start doing human studies is we try it in a mouse. And lucky for us, mice actually get real valley fever infections. They are actual natural hosts, which is really cool. So we worked with partners at Northern Arizona University who ran this, infected the mice for us. And what they did for us is they infected 6 mice with one species of the fungus, Coccidioides possidacei. They infected another 6 mice with Coccidioides immitis, the other species that causes valley fever. And then they had 4 mice that they gave a sham infection, meaning they gave them just the saline that was used to deliver the fungus to the infected mice lungs. And what we found was that these mice, even though they're all genetically identical to each other, they actually had different degrees of infection. So we just measured the cytokines in their lungs, and we can see that the uninfected mice are over here, these white triangles. And then each one of these symbols represents a different mouse that we infected with the valley fever fungus. And we can see all the red circles are representing the mice that were infected with Coccidioides possidacei. All the blue circles are the mice that were infected with immitis. And then again, the white triangles are controls. And we see that on this 2-dimensional plane, that these mice are sort of spreading over this plane. And what we're seeing here is that the mice are separating along this principal component 1, this first axis, left to right. They're separating based on disease severity. So where we see darker colors indicates more severe disease as we measured it by their immune response, by the cytokines that they produced. And these are the most severely infected mice here on the left, all the way down to the uninfected mice over here on the far right. So point number 1 is that in these mice, we saw different degrees of infection. And we measured that by how much fungus was in their body and how much of an immune response they made to that fungus. We also looked at the volatile metabolites, the gaseous metabolites from their lungs. And we found that when we classified or segregated these mice based on their lung chemistry, we saw a very similar pattern where the mice are separating by the severity of their disease. So here towards the right-hand side are the mice with the least severe disease, all the way to those 2 mice over here on the far left that had the most severe disease. Because we see the breath chemistry separating these mice in the same way that we're seeing their immune response separating them, that suggests to us that we'll be able to develop a breath test that not only detects the fungus, detects the infection, but also tells us how severe is your disease. And that can be important for deciding whether or not you're prescribed antifungals. Remember, 60% of people resolve their disease on their own, and antifungals have some tough side effects. It's actually best clinical practice not to give antifungals to every person who might be diagnosed with valley fever. It's better to use them only in people who really need them because their infection is severe. We don't want to treat those people who can resolve it on their own. So from this mouse data, we have indications that we'll be able to measure the chemistry in the breath and be able to detect not just the presence of the fungus, but how much fungus is there and how severe is the disease that it's causing. And now Mitch will tell you about the diagnostics that he's working on.
Okay, one more lesson. This is general biology. Okay, so the central dogma is that DNA ends up making protein. Remember, DNA is all the information of your cell, right? Proteins are the machines and the structures of your cell. And so if you have the DNA that makes one gene makes one protein, that's not 100% correct, but pretty close for now. One gene makes one protein. If you can clone that gene, you can make the protein. And the Moderna and BioNTech vaccine takes a kind of a mid-step. They give you you may have heard of mRNA. That's the mRNA vaccine that they put into your muscles, and then your muscles in your skin, and you make your own protein that is the viral protein. And that's actually what happens in a natural virus infection. Those viral genes get inside of you, and you start making all the viral pieces. So, you know, the BioNTech vaccine just kind of skips that step and gives you the mRNA so that you can make the protein and then make your immune response to protect you. Now, this is Josh's technique. We are so lucky to have Josh at ASU. But up at Harvard, he invented this process. So what I use diagnostics, I want to measure antibodies in patients. And so to measure antibodies in patients, you got to make the protein. But that's not easy to do. But Josh created a way, and it's called NAPPA. It's called Nucleic Acid Programmable Protein Array. You don't have to remember that. Just remember NAPPA, and it's proteins on a microscope slide. And the original version had about 3,000 spots on one microscope slide, 1 inch by 3 inch by 1 inch. Well, luckily, as Josh came here, what I feel is one of my best accomplishments here at ASU is we worked with a team. We got a grant, and we developed the next version of NAPPA. He's already on version 3 or 4. But we developed the second version of NAPPA called high-density NAPPA. And the way you do that is that you create nanowells in a silicon slide. So we worked with engineers, we worked with chemists to be able to create on a microscope slide little bitty test tubes. And we now can put 20,000 test tubes on one slide. It's pretty amazing. So, and what you do, I want to go back. You print the coding region of the protein, and you add a little tag to it. And you add an antibody as a tool, an antibody to the tag. When you make the protein, that protein tag gets captured, and then the protein, the viral or the fungal protein, is displayed for the immune system to see. Then when you add the patient sera, if the patient has antibodies, it binds to the protein. Just like I said, if it grabs on, holds tight, you can wash it won't wash away. Then we take another antibody that has a fluorescent molecule on it to see if there's any human antibodies there. And if you look at this, you get these really pretty images. You can scan these with a special scanner. And you get wherever antibodies are binding, it lights up and it goes from dark blue all the way up to bright red. That's our color scheme. So with binding of antibodies, if you get into the yellows and reds, that's very high binding. And so here we have some control proteins, but I want to point out this right here, this dark blue up here with the healthy folks. But in our valley fever patients, look at that. That is bright red. It's shining. It's saying there's antibodies to this fungal protein. And we can scan this and turn this into digital numbers. And when you do that, we basically have just given this a graph of antibody levels. All of our healthy patients are non-reactive to this protein here. Our patients are anywhere from like 25 to over 100-fold more reactive to this protein. Now, this is one protein that we know is reactive in most valley fever patients, but it's not 100%. So we're creating a protein array with over 700 valley fever proteins. And we want to find, are there new proteins that could be used for better diagnosis? Because the valley fever antibody test doesn't work for everybody. And with that, we're going to kind of stop the data part. I want to acknowledge a few colleagues. Dr. Doug Lake, who's actually housed here in this building, is a co-investigator on my grant to look at finding new antibody targets. He also has his own lab. He's looking at antigens and the fungal antigens in urine and serum. Dr. Tom Griss works with us. He's here at the Mayo Clinic, runs the clinical laboratory and helps get us samples and helps do the specificity tests that we need to do. Janice Blair is one of the chief valley fever clinicians here in the county. If I had valley fever, I'd want to talk to Janice if I could. And
so, just to sum up, the point that we want to really hammer home is that new valley fever diagnostics are needed. The ones that we have are missing a lot of cases. And it's because we're relying on a patient's immune system to tell us that they've seen the fungus. And not every patient's immune system is able to tell us that information. And at ASU, we're taking a multi-omics approach, meaning we're looking at metabolomics, which is what I do, proteomics, which is what Mitch and Doug Lake do. And we are also looking for new antibody targets to help us develop better diagnostics to detect this disease whenever it's impacting patients. And the benefits to patients, I mean, this might seem obvious, but it's faster diagnosis. It, on average, takes people 28 days to be diagnosed. After they first go to their clinician, it takes another 28 days to get a diagnosis for valley fever. So faster diagnosis for patients, fewer invasive tests. You don't have to go in under anesthesia with an endoscope to get a piece of that fungus to try to grow it. That means we're going to have fewer ineffective treatments. You're not going to receive antibiotics, which are not functional at all against the fungus. Instead, you'll be treated with antifungals when you need them. And that's important for antifungal stewardship. Not every valley fever patient needs to be treated with antifungals, which have a lot of tough side effects. But we can target those antifungals to those who will need them most to help recover from their infection. So that's all we have for you today. Thank you so much for having us. We would love to hear your questions and answer those for you. Yes.